This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 441, recorded on May 12th, 2017. Hi, everybody. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Western Massachusetts, Alan Dove.
Good to be here.
How's it going out there?
It's suddenly gorgeous outside. It's 62 Fahrenheit, 17 Celsius, nice few puffy cumulus clouds in the sky, and then it's supposed to cloud over again and pour rain all weekend. Suddenly gorgeous. That's a good name for something. Something, yeah.
Yeah, we're supposed to have a nor'easter here in the Northeast.
Yes.
So you're going to get pieces of that.
I think, yeah, we'll get the edge of it or something.
Right now it's 14°C here, cloudy. It's not such a great day. It's been chilly all week like this.
Yes.
It's supposed to rain a lot tomorrow. Also joining us from southeastern Michigan, Kathy Spindler.
Hi, everybody. It's also beautiful here. It's mostly sunny with a few puffy clouds, 66 Fahrenheit, 19 Celsius.
Also
cool.
Joining us from Austin, Texas, Rich Condit.
Hi, everybody. I got the same sky as you all do, but a few more Delta H's. It's 85 degrees, blue sky, puffy blue clouds. That's 29°C. So here we are.
Yeah, it's almost summer. We're still in spring, right? Summer's not till June 21st officially. Is that correct?
Something like that. 21st, 22nd.
Yeah, that's the solstice.
That's the solstice.
I guess that's when people usually say summer starts.
Well, what else would you do?
Oh, I mean, kind of culturally, it starts around Memorial Day.
Oh, that's right. Memorial Day, yeah, when it's usually raining.
Right.
I want to tell you about the American Society for Microbiology grant writing online course. Learn the best practices in grant writing during the ASM grant writing online course. Participate to receive an overview of the funding landscape. Learn what makes grants successful and receive personalized feedback on your grant. I remember when I did this spot for TWiP, I said, the funding landscape more like the funding canyon. Nothing to say badly about ASM, of course, but that's just the way it is.
Well, that's why you need this course.
Yeah, hopefully. I'm not sure, but try it anyway. You can't lose. You could stay home and take it. It's a 7-part series taking place this fall. The registration deadline is July 15th. You can learn more at bit.ly/ASMGWOC17. bit.ly/ASMGWOC17. And while I'm talking about ASM, upcoming, as we've said a few times on TWiV, is ASM Microbe. That's the big annual meeting of the American Society for Microbiology. We will be doing a TWiV at ASM Microbe. And the program, which has already been printed, and which, if you're going, you will get, has the wrong date because we've changed it. It's not their fault. TWiV will be on Saturday, June 3rd, 2:30 p.m., right down in the poster and exhibition area.
How's that going to work? Won't there be a lot of background noise?
Hold on. It's booth 1737. It's not too bad. It's a pretty big booth, and so we're set back from the crowd. We did a couple of like seminar type things last year in the same space, and it was fine. It's not too much. There's just a little kind of background. It makes it cool. It makes it like you're a lot of people around, right? So it's not so bad. So Rich Condit will be coming, right? Rich Condit?
Yes, indeed. I got my flights all booked and everything.
All right. And we're going to have a special guest. Should I tell him?
I just don't know, but it's so special, you know? This is really special. This is very cool. Sure, why not? Tell him.
All right, we're going to have
Kate Rubens.
Astronaut Kate.
Kate Rubin.
Pox
Awesome.
in space.
Kate Rubins, yes.
That is awesome.
And she, of
That's
course,
excellent.
did some virology. She has papers. She even worked on pox viruses. So maybe she and Rich will bond, But
I want to go to space.
she...
Too old.
No, you can't go to space. But yeah, she'll be our guest for an hour. And it's great because she's there to give the keynote, and we get the opportunity to have her on TWiV, which is cool. And I heard that she actually wanted to do it because she apparently knows about TWiV, which is cool. And I want to find out if she ever listened to TWiV in space. They would be allowed to listen to stuff, wouldn't they?
Sure.
Mm-hmm.
So we'll see. And if not, maybe she would, and that would be cool. The first podcast listened to in space? Maybe not.
No, probably not.
Darn,
So where's this happening? Is this happening at Convention Center?
Yeah, it's the New Orleans Convention Center. There's only one. It's a big thing right by the waterfront.
Yeah, New Orleans is one of the few places in the country that can actually hold a meeting as large as ASM.
Yes, that's why they are limited to certain venues like San Diego, Boston, New Orleans.
Yep.
Those are the ones I've been to lately. All right, so remember, Saturday, June 3rd, 2:30 p.m., and come by. It'll be fun. We don't have it's not as big a room as we've had in the past. It's limited seats, so if you want to check out an astronaut, of course, if you're going to the meeting, she will also be at the, she's the keynote speaker, so you can see her there as well, but it's different.
Yeah.
Up close and personal on TWiV.
Close and personal. It's our first astronaut. Also, the American Society for Virology is going to have its annual meeting a few weeks later, June 24th to the 28th in Madison, Wisconsin, the Monona Terrace, which is their convention center. Friday, May 19th. That is 1 week from today as we record on May 12th. And so that is early bird registration ending. After that, the fees go up, so don't be late. Kathy, I registered, but I forgot which Saturday thing I registered for, and I never got an email. Who would it have come from? Do you know?
I'm not sure.
Who would I ask?
Because I don't deal with the satellites as program chair. We could ask Kristen Bernard. She's the local chair for everything. That might be the best way to go, because she does have a handle on which ones have how many registrants and stuff. She sent that to us a couple times. And that careers one that Glenn Rawl is doing that Rich is going to be on is full with 100.
Yep.
So I think Kristen is the way to go.
All right, I'll ask her because I just don't remember. And usually they send you an email, but I didn't even remember if I had registered. So I went back and I started to re-register, and they said you're already registered. Okay. Yeah. All right. Follow up. Jean-Michel Claverie. Does that ring a bell to anyone?
It does.
Yep.
All right. That was one of the authors of the paper we did last week. Dear Vincent, roaming on the net. Can I say the whole thing in a French accent? That's the first.
Probably best not.
Roaming on the net. We discovered yesterday that you dedicated a whole episode of TWiV 440 to Numea virus. Thanks for contributing to our fame. Notice he says contributing to.
Right.
Not for making us famous, because obviously he's already famous, right?
Right.
Even though you made fun of me and Chantal about our style, strict embargo, circumvolution, etc., we were impressed by the details in which you went through the paper. Good job. I wish our students' postdocs would go into such details too. By the way, did you like the circumvolution image?
Yeah, that was good.
I just searched. I said, what the heck? Let me search for circumvolution. And up came a piece of art that embodies the word, I think. Kathy, did you like it?
I forgot to look. I'm going there now.
I knew it, because usually you're the first to say I love it.
I would have commented on it, yeah. Oh, that is way cool.
Isn't it cool? I like it.
Yes.
Yes. It is. That's a circumvolution.
Yeah. That's really neat.
All right, continuing. However, some of the questions you asked were actually answered in the ... supplementary materials, such as messenger polyadenylation. As for tracking the viral DNA inside the host cell, we tried using EDU, but had no success yet. Same in expressing other GFP-tagged proteins. This is unfortunately tricky in the acanthamoeba system. As mentioned by Richard Condit, such experiments have been attempted with pox viruses for dozens of years without success, so give us more time. As for the recent paper on the new, quote, supergiant, unquote, Klaus-Nu viruses authored by Schultz et al. With Kuhn, I and others have huge reservations about it. And I will probably post my comments about it on PubPeer and or PubMed in the near future.
Emphasis added.
Basically, I don't believe in viruses until I have isolated in a tube. I believe ICTV does agree with me in not taking for granted metagenomic assemblies as good enough evidence for new viruses, especially revolutionary ones. Thanks again for your interest in our research. Bien amicalement and meilleur souvenir. Jean-Michel and Chantal. Now, let me just make two comments here. First, supplementary materials, I hate them. I hate them, I despise them, and I do not usually look at them because they belong in the bloody paper. And I'm sorry we didn't see that, but that's why. Second, we never had a T-Rex, yet we classified it. We never had a living T-Rex or a Velociraptor or any of those. We classified them. So why not? That's what Jens Kuhn said anyway. I always steal that because I love it.
Right.
So, yeah, I have a couple of comments here. I looked through the supplementary materials, and my basic question is, are the RNAs polyadenylated? Mm-hmm. And I couldn't find anything in the supplementary materials that addressed that, though I can't say I read them in excruciating detail. I looked over them pretty carefully. My other question was, where does the poly-A polymerase come from? Well, that's not really as critical a question because if it's accessing nuclear proteins, mm-hmm. Either by leaking out or by an alternative mechanism of actually spending some time in the nucleus, then the poly-A polymerase can come from there. But yeah, so that's not as much of a question. I was just wondering whether the RNAs are even polyadenylated. They must be. At any rate. Yeah, and I guess he's talking about reservations about the Klaus-Nu virus just because it was put together with a metagenomic assembly. I don't know whether he's also talking about the fourth domain of life thing and the impact of the Klaus-Nu viruses on that. And I just wanted to point out, as I think we did before, that the idea of a fourth domain of life has been pretty rigorously challenged already in a publication from Kuhn some time ago that I put a link into here.
Yeah.
That's we discussed it before from 2014. So in my most humble opinion, that whole fourth domain of life thing is on pretty shaky legs because I think that the phylogenetics is pretty good for showing that the genes carried by these viruses can actually be traced to existing life forms. And we can talk a little more about this after the next letter, which I found really cool, and I'm glad this guy wrote.
I think, Jean-Michel, you should send your comments to us as well. Because I got to tell you, more people will hear it here on TWiV than
we'll read it on PubPeer. Although on PubPeer, it's going to get directly to the authors almost immediately.
That's fine, but you have to get with the new way of communicating. And this is these are people who really are interested. You can also send it to PubPeer, but why not send it to us?
Do both.
But you won't be listening because it's, as you are roaming on the net, you won't find next week this conversation. So what can we do? I wanted to say something else here. One more thing. Oh, so he's basically saying he doesn't believe that this virus exists as assembled from metagenomics, but this is what people do all the time now. If we didn't do that, we wouldn't be missing a lot of our conclusions, right? So I'm not sure you could ever. I guess you could try and recover the virus from the sequence, but it might not be exactly what the virus is.
Right, but he raises a good point that these new viruses that we're discovering only from sequence. I mean, you've certainly talked about how PCR product is not a virus, And there is some concern that if you have, especially if you're trying to argue that there's an entirely new category of viruses, I think you need a little more than a sequence to go that far. Well,
I think Jens has said that they are considering moving forward with just sequences, if I'm not mistaken. Anyway, Rich, why don't you take the next one since you like it so much?
Okay. Near writes, hi all. I just finished listening to TWiV 440 where you discussed the transient disruption of the nuclear integrity by the giant pneumovirus. Rich mentioned that he thought the evidence was not conclusive as to rule out the possibility of the viral DNA first entering the nucleus to initiate transcription. This reminded me of the entry process of two small DNA viruses, SV40, a member of the Polyoma viridae, with a capsid of 50 nanometers, and MMV, minute virus of mouse, a member of the Parvoviridae with a capsid of 26 nanometers. That's way small. Both of these viruses transiently disrupt the nuclear envelope integrity during their entry into the nucleus. Similarly to what was described for pneumoviruses, these disruptions consist of transient morphological changes in the nucleus and increased leakiness. I've attached the relevant papers. Keep up the good work. Near a postdoc in gloomy Chicago where the weather is currently 7C, cloudy, and nowhere near as nice as back home in Israel. And he gives two links to two different papers, one for the parvoviruses and the other for SV40. And I had a look at both of these, and they're, I think, in the context of this discussion, fascinating. Now, these are both small DNA viruses that do replicate their genomes and do all their transcription in the nucleus using nuclear enzymes. So the genome actually enters the nucleus. And what's shown in these papers is that for a period of time, fairly early during an infection, presumably while the virus is getting its genome into the nucleus becomes misshapen and it loses some of its integrity. In this particular case, assayed by lamin, which is a nuclear protein, a nuclear envelope protein, coming out into the cytoplasm. So here we have, and this is what was shown in the ChrY paper for the giant virus, is that the nucleus becomes misshapen and stuff leaks out. So the point here is that is not necessarily a phenomenon that's specific to those viruses and can even happen with viruses that replicate in the nucleus. Now, the same experiments, it's been shown by numerous methods that stuff leaks out of the nucleus during a poxvirus infection, but I've never seen experiments done that are quite like this to show the nucleus, assess whether the nucleus loses its shape and generally whether proteins leak out. And I think that would be nice to see in the poxvirus infection as well. But at any rate, going back to the paper that we discussed last week, we got two observations. One, that the virus doesn't have the equipment in the virion that it would need to establish a replication in the cytoplasm. And two, the nucleus becomes misshapen and stuff leaks out. The conclusion that was drawn is that some of the stuff leaking out is probably transcription stuff that helps initiate the infection in the cytoplasm. But I think they ought to do some more stuff, okay? Because what is missing is the idea, as happens in iridoviruses, that the genome could enter the nucleus transiently and start replication and transcription in there, and then some of the replicated molecules could exit the nucleus and establish the factories in the cytoplasm that people see. And nowadays, there are techniques that could be used to look at this pretty well. What comes to mind, what I would entertain doing, is RNA scope. Do you know about this technique? Mm-mm. This is it's basically an in situ hybridization technique, but it is extraordinarily sensitive.
Yeah.
And works really well. And I don't see why that couldn't be used for looking for DNA as well. So you could look for specific early or late genes and specific for DNA replication and look to see whether it happens in the nucleus or the cytoplasm. I also looked up the old iridovirus papers, because the iridovirus thing, where it spends some time in the nucleus, initiates replication in the nucleus and then finishes off in the cytoplasm. That is, the DNA molecule enters the nucleus, at least this is the story, enters the nucleus, does early transcription at least, does a round or two of DNA replication, then the DNA is exported to the cytoplasm, where it undergoes a lot more DNA replication. You set up cytoplasmic factories. It's not actually clear where late transcription happens. It hasn't been studied well enough. But those experiments that lead to that model are 40 years old. They were done in actually even more than 40 years old, in the 1970s, doing autoradiography, where you label either RNA or DNA in cell culture in infected cells with tritium, and then on a slide cover it with an emulsion and stick it away for 2 months and look for tritium grains, exposure of the emulsion over either the nucleus or the cytoplasm. And those are the types. They also did some subcellular fractionation. Maybe those experiments ought to even be revisited. But this is a really interesting problem to me, and I'd like to see it investigated further. And I hope they do.
It sounds like they're working on it.
Yep.
It's cool. All right, Alan.
Trudy writes, Dear TWiVers, I'm writing this letter in response to Allison's letter on TWiV 440 regarding the adverse effects of vaccination. Since Alan seems to think that anecdotes are more likely to attract the attention of people who are opposed to vaccination, I would like to offer a few of my very own personal anecdotes. Anecdote number 1. I had severe cases of the flu 2 years in a row in 1998 and 1999. I remember those years exactly because the illness was so debilitating that I remember wishing for nothing more than good health. Both times, the illness lasted a total of 2 months, mostly due to the bronchitis that developed as a secondary infection. I will never forget the misery, the pain, and the feelings of hopelessness and uselessness because I just wanted to feel healthy and productive again. Since 1999, I have been getting a flu shot every single year, and since then, I have not had the flu. I have been around plenty of people who had the flu, especially family members and coworkers who refused to be vaccinated. This year in particular was a very difficult flu season, with lots of coworkers missing work repeatedly for extended periods of time. I, however, still did not get sick. If I did get infected with influenza virus in the past 18 years, the illness manifested itself with minor aches and pains, which maybe lasted a total of 1 to 2 days, and after which I felt completely healthy again. This is consistent with reports indicating that even if the flu vaccine fails, in most cases the severity and duration of the symptoms are significantly reduced. Sorry, I used a statistical term. I just can't shake the scientific approach. Anecdote number 2, my children, who have also received the flu vaccine every year of their lives, including during their in utero season, have never had a full-blown case of the flu. If they did get sick, the illness lasted a total of 1 to 2 days, and they never developed any respiratory complications. In addition, they are, at their respective ages of 1 and 3, completely up to date on their vaccines, and they've never had any adverse effects from their vaccinations, besides the slight elevation in body temperature that sometimes occurs the night following vaccination. They've also never had any other serious infections. They've never had measles, mumps, or rubella. They never had polio, diphtheria, rotavirus infection, hepatitis B, pertussis, tetanus, bronchitis, pneumonia, or chickenpox. And because they haven't had any of these diseases, they haven't passed them on to other children. You know what else they haven't had? Smallpox. And you know why? Because smallpox was eradicated with a very effective vaccine more than 35 years ago. Anecdote number 3, I did not receive the MMR vaccine as a child, and consequently I had a full-blown case of the mumps at the age of 7. I remember the illness distinctly, as I was bedridden for at least 2 weeks with extreme pain in my salivary glands. I specifically remember the pain as I tried to lift my head off the pillow, which you have to do every time you turn in bed or get up to eat or go to the bathroom. Even though I obviously eventually got better, this disease left me deaf in my left ear, 85% deaf to be exact. While it is not the end of the world, I'm perfectly healthy otherwise, 85% deaf in one ear means you can't hear sound in stereo. So you often don't know which direction sounds come from, such as elevator dings, people calling your name in crowded places, car horns, etc. You're also a nuisance to the people around you as you're constantly asking them to repeat themselves. You listen to music and audio really loudly, once again annoying those around you. You always watch movies with subtitles on in order to keep the volume at a reasonable level so as not to annoy those around you. You may not hear people when they're talking to you from the affected side, especially in environments with the slightest background noise. If people don't know you or about your disability, this can create some awkward social situations, or some social awkwardness. Whispering anything into that ear is completely out of the question, as is telephone use on that side. Long story short, it is a major, major nuisance. It may even be a hazard at times. What I've noticed with this entire anti-vaccine sentiment is a very unfortunate glorification of the old days, because life was allegedly so much simpler and less toxic back then. But that is not so. People routinely lost children to simple bacterial infections. Women routinely died in childbirth. Surgery of any sort was always very risky. My own distant family history is rampant with infant, child, and adult deaths from pneumonia and complications from various other bacterial infections. Why do we forget that? Maybe we need to remind people about science history. Anyway, keep TWiVing, dear TWiVoners.
Oh,
yeah.
Good.
Great stories.
Yeah.
We were just talking about this.
Multiply that by millions.
Yeah.
That's,
I use her anecdote number 3 as one of my anecdotes because I have 3 friends. She's one of them who have been affected because of not being vaccinated.
Yeah.
And in her case, it just wasn't available where she lived.
Right.
All right. So people who want explanations for things that happen to their kids, it's not vaccines. I know you want to hold on to something, but vaccines are good for other things. They don't do all these other bad things.
There is such a thing as a vaccine side effect or a vaccine reaction. These things do happen, and they're well documented in the scientific literature, and that's why not everybody gets every vaccine. There are some things, we don't just go around willy-nilly vaccinating everybody against, rabies. It's an effective vaccine, but it's not great, and it's not long-term effective, and it's a rare enough condition. So, the risk-benefit analysis favors not bothering with the vaccine, especially since it's effective after exposure, and you probably know when you've been exposed. But for these other conditions, really makes a lot more sense to get the shot.
These childhood illnesses that Trudy mentioned, get your kids vaccinated.
Absolutely.
One more.
Yes.
Kathy.
Jeffrey writes, I guess one could say that the AHCA bill is the kind that makes you sick. So the AHCA bill is the one that the Republicans tried to get passed, and it failed.
They just passed it in the House.
So it kept the same name?
Yeah.
When they revised
it's certainly
it?
being referred. It's the same bill.
Okay.
They just actually made it even worse, and then they got apparently enough people to sign on because they wanted to be meaner, and so they managed to get that passed. And yes, that is the kind that makes you sick.
Great play on our little tag.
Yes,
for sure.
Yes. And reminder, it's only passed in the House. So for those of you that might suddenly be worried, it would have to pass the Senate.
And for those of you whose senators have not already come out strongly condemning this thing, this monstrosity, which if you haven't read it, it's not actually a very long bill. It basically robs huge sums of money from the poor and gives it to the wealthy. And I'm not actually caricaturing it by saying that. So you can contact your senator and let them know how you feel, unless your senator is already all up in your Twitter stream telling you that they hate it.
Many do.
Oh, yes. So this is a very, very unpopular thing.
Many weird things happening these days.
Oh, yes.
All right, let's do a paper. Because that's what we're here for on TWiV. And this one is published recently in the Proceedings of the National Academy of Sciences. It's called Genetic Variation on Chromosome Y Regulates Susceptibility to Influenza Virus Infection. And we have a first author of Dmitry Krementsov, followed by Case Deans, Raza Fang, Aether, Pointer. Hey, a pointer.
Mm-hmm.
Same spelling.
Mm-hmm.
Boyson, Bunn, and Corey Teuscher from the University of Vermont in Burlington, where I've been once in February. It was very cold.
I bet that is a cold place in February.
very cold, very bitter.
I was up there in summertime once. It was gorgeous.
It's lovely in the summer, yes.
Yeah.
All right, influenza A virus, as we have heard, is a big problem. Infects many people every year and has caused multiple pandemics. And looking at the data, epidemiological data, it looks like the morbidity and mortality, so morbidity being the disease and mortality, death, of course, are greater in women than in men. So this paper is all about sex differences in susceptibility to influenza virus. But if you look at the data as a function of age, it looks like more males are affected from birth through age 15, and then beyond that, the women are more affected. So it's not as simple as saying more significant in women than men. You have to stratify by age. And emerging data are suggesting that genes on the X and Y chromosomes may influence disease susceptibility. Okay, and there are a couple of studies that suggest that. For example, Coxsackie B virus seems to be controlled by genes on the Y chromosome. So what they have looked at in this paper is whether in a mouse model of influenza virus, they have a mouse-adapted strain called PR8, commonly used, we've talked about it before, intranasal inoculation, whether the Y chromosome makes any difference. And they have a really interesting panel of mice. So, as you know, there are lots of different mouse laboratory inbred strains of mice with names like Black 6 and Lewis and AJ and et cetera, et cetera. And what they have here is a panel of mice on a Black 6 background that have a different Y chromosome. These are called consomic strains of mice.
Right. So they have 11 of these different strains, and they have Y chromosomes from 11, I guess, other strains of mice, right?
Yeah.
Mm-hmm.
But Alan,
B6 is one and 10 different other inbred strains.
But why?
Why?
Yes, why? Why not?
So, the consomic, I'm sorry, I didn't know that word before. I looked it up. It's a great word. So basically, just one chromosome is different? Is that what consomic means?
That's what it means to me. All of the other chromosomes in these mice are the same. That's important. So the only difference from one mouse to another is the origin of the Y chromosome.
How do you make these kinds of mice? Does anyone know?
I don't know, but I would imagine that it.
I assume it's done through breeding.
Yeah, you start with
you have to breed. But
B6
how do you just randomly screen through the offspring and find the one you want?
I
would imagine, that there's a bunch of that you would cross B6 with some other strain,
right?
And
so
then
you cross.
if you take the F1 and back cross them to B6 indefinitely.
Yeah. So if you start with a B6 with the B6 Y chromosome background, you take females from that strain, right, and then you breed them with males of some other strain, and you're looking for transfer of just the Y chromosome. So then you take the males from that, and you could design a set of crosses that would eventually accomplish this.
So you do have to screen because the males are going to get other chromosomes.
You do, exactly, because they're going to get other things.
So I wonder how you do that, yeah.
Googled for how do you make a consomic strain, and the first ones were made in 2004. If I read that correctly, Joe Nadeau, Eric Lander, and coworkers, I assume it's Eric Lander, made the first set with the AJ strain as a chromosome donor and B6 as a recipient strain. That was published in 2004. And when I bring that up, Eric Lander is one of the authors. And so 65 other strains have been made. And so the transfer of sex chromosomes was not reported. And they... To get at how they do it, sorry, I'm not able to parse it quite quickly enough. This paper
typically what you do is you take the F1 from a cross and you back cross it to the strain that you want everything in repeatedly. So you take a B6 and some other mouse, cross them, get the F1 from that, take the males. You probably do a screen that involves taking a tail snip and doing some sort of, I don't know if you can distinguish the Y chromosomes by karyotyping or PCR or a little bit of both, but at any rate, you screen them actually probably by microsatellite or something like that, but you'd want to make sure they got the whole thing. At any rate, you take guys that have the Y chromosome that you want, and you back cross them to B6. Okay, you take the guys from that, you back cross them again, you back cross them again, you back cross them again. Screening at each stage. I'm making this up, but I've seen this done with other things.
So basically, it's not that different. What you're describing isn't that different from how you'd make a congenic strain, which is a portion of a chromosome. Right. And here, the consomic, the some comes from chromosome. Right. And so what they say in the introduction is it was created by transferring segments of individual autosomes from one strain into the background of the other, and then that's where I don't exactly see their details. So I'll continue to look at that.
Yes, you could do it in a more modern way, couldn't you? You could, just in vitro transfer this, couldn't you?
Well, that's what I was wondering, if there is some other way to do it, but
do it more surgically.
But it does seem to be the more typical back-crossing way.
Okay, so we're not entirely sure how these strains were developed, but the upshot is that they have these 11 strains with Y chromosomes from different other strains of mice.
I don't think it's trivial. I think it takes a long time.
No, I think this was a big thing to get.
And I think this is a reagent. I'm not sure that if they made it or if they had.
No. They got them all from Jackson Labs.
Jackson has them. You can buy them.
Because I was asking this question of myself and looking through the methods, and what they say is that they got them from Jackson Labs.
Okay.
Right.
Yeah, but who deposited them into Jackson Labs?
Right.
Exactly.
That's who must have done this. The hard work of getting these. Yeah.
Right, and it might still be the Neto and Lander group. The other point I wanted to make is that because they're not only are they all the same with respect to their autosomal and their X chromosomes, those, because they're inbred mice, they're completely identical. So we're not talking about outbred mice with differences in all their autosomes. They're completely identical.
That's right.
Like
so
twins.
They say when they got these that they back-crossed them against female Black 6 mice for an additional 5 generations before they did the experiments. So that implies that this back-crossing protocol is part of the generation of these things, and they just did a little more to make sure they were absolutely clean. And it occurs to me that in making a consomic strain, that if you take a cross and isolate the males, by definition, they're going to have the Y chromosome, the whole Y chromosome from the male you started with.
Yes.
Okay? And if you're back-crossing those each time to females, there's not going to be an opportunity to get some sort of hybrid Y chromosome. So if you keep doing those back crosses indefinitely, you're going to wind up with something that is consomic. It's got the clean whole Y chromosome, and the others entirely whatever the genotype strain is, the background strain that you're back crossing to.
And just if you're wondering how long this takes, so the gestation period from a mouse is 21 days, and then I think between 2 and 3 weeks you can mate them again, right?
You usually mate them at about 5 or 6 weeks old.
But you can actually do it 2 to 3 weeks, I think.
Yeah, but
so a month minimum for a back cross. So this is 6 months to do it. What did they say? 5 back crosses, 5 to 6 months.
Yeah.
I mean, if you want to accelerate it, you'd want to mate them earlier than 5 to 6 weeks.
Right, and then there's the screening in between. So you might go ahead and mate them further, but you would be screening to make sure that they that you're mating the ones that you want.
Yep.
And that takes time.
I know, because we used to separate mice. You would wean them at a certain time, and sometimes they would already be pregnant.
Right.
So that was precocious mice. All right, so then they take these animals and they infect them with influenza virus. 5,000 egg infectious units, which is 1 LD50 for the Black 6 mouse. Okay, so 5,000 egg infectious units will kill half of Black 6 mice. they put it intranasally, they put it in all these strains, and then they do a survival curve. And interestingly, these 11 strains segregate roughly into two general areas of susceptibility. There's the ones that get about between 60 and 80% survival, and then the other group between 5 and 30% survival. Two clear groups, wouldn't you say? Would you agree that there are two clear groups of survival?
Yes.
Right? And these all, so the idea being that the ones that are more susceptible have different Y chromosomes from the others. So this is segregating with the Y chromosome because the rest of the chromosomal complement, as we've just said, is the same. Two groups. And this survival doesn't correlate with weight loss. Now, one of the things that happens when you infect mice with PR8 is they get sick and they lose weight. And they say here that when you look at the data and you include weight loss, the survival is independent of weight loss. Now, they wanted to know what is the difference between this and susceptibility of female mice, because previously it had been shown that female black 6 mice are more susceptible than male mice. And so they add in female black 6 results to the graph. They've now put it into two groups, the high survival and the low survival group. And the black 6 males are in the high survival group, and then a bunch of the other chromosomic strains are black 6 male-like. They also survive well. And then you have that other group which have lower survival, and the black 6 females fit into that group. So that is why there was this previous, or partly why there was this previous idea that females are more susceptible because the black 6 females are, but that's just they're just down there with the Y chromosomes that confer lower survival. Okay, I think that's pretty cool.
Right.
Mm-hmm.
So it's looking like there's something on the black 6 Y chromosome and some other the Y chromosomes of some other strains that is maybe protective.
So what we should just do is give the female mice a Y chromosome and they'd be protected.
Yeah, they'd also be male.
Oh,
Right.
I forgot about that.
There's that.
Right. Or maybe just the genes from the Y chromosome.
That's yes,
Maybe
or
we
the
could-
effect of the genes. That's
Yes.
what we're really getting at here.
So what is going on? This is the really interesting question in terms of the virus and, of course, the Y chromosome. So the mechanism, the first thing you would look at least I would, is how much virus is in these animals. Does the amount of virus correlate with survival? So they look at whole lung viral RNA, unfortunately. They don't do plaque assays. And they see no difference between the highly susceptible and the resistant mice in terms of viral replication. Increased mortality is not due to enhanced viral replication. I would like to see a plaque assay, though. I really would. It's not that hard to do, folks. Not hard. I used to do them all the time. Many people do. Who knows? Maybe there's just something weird happening here, right, with the RNA. You just don't know. I mean, someday we're going to find that this is a big problem. All right, to move on, what's going on in terms of the genetic response of the host, the transcriptome, the RNAs that are produced? All right, so you extract total RNA and you sequence it, you amplify it and you sequence it, and they find 32 genes. So they're now looking at very highly susceptible and less susceptible representative animals.
Yeah, so just one strain of each. One of these 11 strains, that is the highly susceptible one, and then the B6 as the control.
The B6, which is less susceptible, right? And the one that they use is a PWD strain of mice, right? 32 genes are higher in the PWD lungs compared to black 6 lungs, and 98 genes had lower expression. And it's very interesting. A minority of the genes with decreased expressions were coding. The rest were classified as small RNAs. So these are the ones that are going down in the highly susceptible mouse lungs. So that's interesting. Go ahead.
Yeah, not just non-coding, but small RNAs, ribosomal RNAs, a little bit of microRNAs, and some of the coding RNAs also go down. But basically, the majority of things that are going down are the small RNAs.
So they
those small RNAs include a lot of things that we would now suspect. I mean, somebody says small RNA, and I'm thinking of this whole regulatory network of RNA molecules that we now know exists in cells. So there could be this could be an indicator of some sort of a complex gene regulatory program that is being influenced by this chromosome.
Exactly. So the idea may be that it's not on the Y, the actual gene is not on the Y, but it's on another chromosome that's being regulated by something on the Y chromosome.
Right.
And in fact, none of these genes that they measured, up or down, is encoded on the Y chromosome.
Right.
That's right. Now, several genes that went up in these susceptible animal lungs were of immunologic relevance. Two of them encoding a gamma delta T cell receptor, some immunoglobulin genes, and they say that the gamma delta T cells genes are interesting because there's an association between those kinds of cells in lung injury caused by influenza virus infection. So they pursued that. They isolated cells from infected mouse lungs, B6 and PWD 6 days after infection. And they do flow cytometry, and they found a twofold increase in the number of gamma delta T cells in the lungs of the more susceptible mice compared with black 6.
With appropriate control showing that other sorts of T cells are the same in both animals.
That's right. No difference in the proportion of B cells in the lung, which, of course, are the antibody-producing cells. There are a number of subsets of gamma delta T cells, and they go through some experiments that show a specific subset of these gamma deltas are the ones that are going up. They also show increase in a cytokine interleukin 17 in the lungs, and this is produced by these gamma delta T cells, this IL-17. And IL-17 is a pro-inflammatory cytokine, causes inflammation, which can cause damage. So they're wondering if maybe this increase is in part causing the damage or the more severe disease in these animals. So it could be immunologically based, because we already saw that it's not an increase in virus replication, so that would make a lot of sense, right?
And we know from pathogenesis work on flu that the immune response is a huge part of the problem.
It is. So that brings up the immediate solution to flu is just take out your immune system.
Take out your immune system. Then you don't, well, yeah, there's some other problems
There's
there.
some other issues with that. All right, next question. The current paradigm is that sex differences in susceptibility to flu is a consequence of the immunostimulatory effects of 17-beta estradiol and the immunosuppressive effects of testosterone on the immune system.
Right. So I wanted to make a little aside here, and that is that for a number of infectious diseases in many organisms, the males are more susceptible than females. So that's the opposite of this influenza situation, except that it's similar to the situation for influenza males up to age 15.
Right. And again, the testosterone theory would again fit neatly, if true, with this idea that the immunological response is a big part of the problem with flu pathogenesis. If testosterone is tamping down the immune response, then you would expect in a disease like flu, the males might actually come out better. But as we're about to see, that may not be the answer here.
However, Alan, this again points to a solution to flu: just get rid of testosterone.
Just get rid of testosterone.
And many other problems as well.
It may solve many other problems, yes. It would have some side effects.
So they do measure testosterone in their mice, and they correlate that to survival. They do see an effect of the Y chromosome on serum testosterone levels, which I love. They abbreviate capital T.
Right. Even though in the paragraph ahead they had T as T cells.
Right.
Yes. Why wouldn't you just write out testosterone, right?
Or use, but I think that's the standard abbreviation for testosterone. But they don't have to do the testosterone experiment here on these mice. They've already done that and published it. This is the first of many experiments in this paper where they're bringing past data to bear. They take their susceptibility data that they generated and they're comparing it to all these other things that they've measured in these same consomic strains and published.
But couldn't it be that the infection has an effect on these levels, right? Because their past data are from uninfected mice, right?
Right.
Right. I know that the results say that testosterone levels are not associated with survival, but who knows if the virus is affecting that or any way.
Right.
Anyway.
But then they refer to Sabra Klein's work, which does talk about testosterone in the context of the influenza infection, and they say that the results are similar.
Okay. And she, of course, handled this paper for PNAS.
Mm-hmm.
So she made sure it's right.
Yep.
So testosterone levels are not likely to be major drivers of differential susceptibility in these variety of consomic mice.
So that very appealing theory that testosterone was driving this effect is pretty much gone.
So we can leave testosterone in. We don't have to take that out. But
not for this reason.
We could, of course, engineer people not to have testosterone, but then they wouldn't be males.
Right.
And even as females, they wouldn't be quite right.
Even as females, they wouldn't be quite right. Yeah.
All right. Now, the other thing they look at with testosterone is whether levels during development, because these are the levels we just talked about are adult levels, so they want to see if there's a developmental effect of testosterone on this. So they look at that. Now, Kathy, is that also previous data? Developmental testosterone, and they regress that
AGDI index. Yeah.
And
that's published in reference 42.
So differential susceptibility is not a function of differences in testosterone during development either. So I guess we can put capital T to rest for now as far as flu goes. There are a number of then the question is where on the Y chromosome is having this effect, and there are a number of alleles that they know about, and I guess again they have previously studied these alleles in these various mice, right?
Mm-hmm.
Like SRY, sex-determining region of chromosome Y, is one of them that they look at, and that this does not correlate with susceptibility. So that's one. I think that's the only one they look at here, right? SRY.
In this paragraph. Yeah.
Then the next one is multi-copy genes on the Y chromosome. So the mouse chromosome Y has several multi-copy gene families, which have previously been shown to be associated with experimental allergic encephalitis and Coxsackie virus B3-induced autoimmune myocarditis. So they look at some of these genes here as well. None of them correlate with differences in survival following influenza virus infection.
Right. So what they're doing at this point is they're casting about at the things that most obviously could be causing this and gradually eliminating one after the other very attractive candidates.
Yeah.
Right. And this one was particularly attractive because in the case of Coxsackie virus and the experimental EAE, that those did show differences between the different sets of Y consomic mice.
Yep.
Yes, this was a very sensible thing to go after, and turns out that's not the answer either.
And I just wanted to add a little aside about copy number variation, that most genes on the Y chromosome, if they're present in one copy, are going to be, I forget the exact term, but basically haploid with respect to their gene expression compared to something that's on an autosome, for example. But if there's multiple copies of it, then that might be a way of getting, more copies within an individual.
Yeah.
Just a little odd aside about Y chromosomes.
They then do an experiment with Coxsackie virus B3 to see if they can see an association between susceptibility to that viral. That's of course a coronavirus and some of these multi-copy genes.
Some of the specific ones, yeah.
This is SRY, SSTY1. So there's a whole bunch of them that they look at, and including RBM31Y, RBMY, they're RNA binding proteins, SLY, SLY, which I like, SRSY, SSTY, and etc. And their studies revealed that SSTY, which stands for spermiogenesis-specific transcript on the Y1, so these are things they don't know what they do, but they're there. This is associated with survival to Coxsackie virus B3. So,
but not flu.
Not flu.
Right.
Just Coxsackie virus B3. So this is a virus-specific thing.
Right. So the take-home message is that the chromosome Y genetic variation underlying the phenotypic differences in survival for flu are different from those for Coxsackie virus.
Yes.
All right.
And don't correlate with hormones.
Right.
And don't correlate with a lot of other stuff. They do.
Right.
The one thing they notice is this increase in gamma delta T cells
that make IL-17. Right.
Right.
Doesn't correspond with replication apparently either, as assessed by amount of RNA.
Right.
So some identified chromosome Y variation, not linked to copy number genes, is important for flu. And they say to really get at this, we have to sequence the short arm of Y chromosome from each of these strains, which is 3.5 million bases, which is nothing. It could have been done by now, right? That's nothing.
Well, maybe that's the next paper.
They must have already done it.
Well, and let me look at the strain names. Yeah. Well, I know obviously B6 has already been sequenced, and I don't know if that particular substrain of 129 has already been sequenced in entirety, but I would bet that, it wouldn't be that hard. But
I sense a bioinformatics paper coming.
Right.
Well, this group has published a number of papers using this same set of mice to do related experiments. So it would behoove them to have those molecular data to. So
the idea would be
help evaluate what's going on,
to see if you can narrow it down to a single gene, right?
Yeah,
or a couple of genes, and then figure out how they work, right?
Right.
And of course, they may not. They may be their target may be in another chromosome, as we said earlier. Yeah.
So I want to visit something here for both myself and for the lay folks out there, and that is about the role of the immune response in morbidity in infections. So obviously you want to have an immune system to fight off an infection and to help you remember that you did that so that you can be immune next time around. And I think it's worthwhile pointing out that a lot of the symptoms that we see in a lot of infections really are the immune response, not the infection itself, a lot of inflammation and stuff. But as I understand it here, there are circumstances where the immune response kind of is too much and it causes a problem. Is that a fair statement?
Yeah.
Yes.
Immunopathology. Yep.
Okay. So in the one of the bottom lines here is that it's important, for an optimal immune response to have the immune response tuned very finely to the infection, and that's a complicated issue.
Yes.
And it's controlled genetically, and there can be circumstances where the genetics is such that balance is sort of results in sort of an overactive immune response that ultimately, to the person who's been infected, causes a problem.
And the immune system is prone to overreaction, and you kind of want it that way, because if it underreacts to a pathogen initially, it may be too late to correct that later.
Okay.
Yeah. I mean, it is regulated, but that varies in different people, and it varies in response to different pathogens.
It has to be extreme enough to eliminate or contain the threat, and that's going to have side effects. And those side effects are actually a lot of the symptoms that you suffer when you get a cold or when you get the flu. That whole crummy feeling and the congestion and everything, that's all that's all the cytokine storm and the immune response that is reacting to that threat.
I would say there are very few, if any, human viral infections that don't have some component of immunopathology.
Yes.
It's just a lot. And I think if you think about what the immune response is doing, you're making cytokines, some of which have cell-damaging properties. They pull other immune cells into the infected area. You get swelling, you get fluid coming out of blood vessels. It's bound to be some damage. It's damage, collateral damage, I guess. Right?
Yes.
Yeah. After all, some of the immune responsive cells are cells that are designed to kill other cells.
Mm-hmm.
Yes.
Right.
Well, in Coxsackie B myocarditis in mice, right? You infect mice, the heart gets trashed by the virus. If you take out the ability of T cells to kill infected cells, there's no damage to the heart.
Okay.
It's all caused by cytotoxic T cells. That's probably an extreme example, but
still, that illustrates the issue.
Yeah.
Right.
So the bottom line on this paper then would be that there are sexual differences in the sexes responding to influenza virus infections, and by the way, other infections as well. In this particular case, males experience more morbidity and mortality, and these guys are pursuing that in a mouse model to find out what it is about the Y chromosome that makes you male that would influence this. And they're able to reproduce the differences in a mouse model, and with different Y chromosomes, they get different magnitudes of effect from nothing to a clear preference. And the only thing it tracks with it is this immune responsiveness that in the cases where the mice are experiencing a more morbidity and more mortality to the influenza A, they're getting a more robust, a more pronounced immune response, specifically with gamma delta T cells in the lungs. So it suggests that the difference has to do with the immune response, and it also suggests that the Y chromosome, probably indirectly by tweaking gene expression elsewhere in the genome, is influencing the immune response. I got that right?
Yeah, that's right. And understanding this is important, because if you can find out why some men are more susceptible to an infection, maybe that helps you design ways to prevent it. Now, if you're worried about this being in mice, they do cite a study in humans where chromosome Y seems to control progression to AIDS in certain men. And so you can clearly do these kinds of studies in people. And so now, having this result in mice, you can go out there and ask, okay, in men who get severe flu, what's the difference between them and men who get less severe flu? Right?
And we talked about some of the human data up top too, with women being affected versus men in different age ranges. So
and now you could go in and get some blood and look at their chromosomes, right?
Right.
And see if you can put them into certain groups, haplotypes, as they say, and then go all the way down to the gene level. So this is the kind of thing you could do in people, because people get influenza. You don't have to infect them, and you can get some blood from. All you need is blood, to get out some chromosomes.
But you need to get at the mechanism in a tractable system like mice, where you can give them all the same virus at the same time and back cross them to get pure strains and all that. And then when you've got some candidates, you could go into humans, look at the homologs of those genes and say, what's going on here?
Well, here it tells you it's chromosome Y, right? Because if you just went for the human data, you'd say, okay, well, maybe it's Y because it's men versus women. But
let's sample a few billion people and find out. That's going to
right. Exactly.
Try and get that grant.
I love this picture of the Y chromosome. Who put that in here?
I put that in there.
That's good.
Some of the genes that have been mapped to the Y chromosome include channel flipping.
Yeah.
That's the FLP gene, catching and throwing, ability to remember and tell jokes. I've got a mutation in that gene.
Got one gene. Yeah.
Ability to identify aircraft. Yeah, this comes from at least sometime in the 90s.
I think
not
so.
The 80s.
Yes.
Yeah.
For a period of time, I taught genetics to the incoming graduate students in our sort of general class, and I always used this.
Pre-adolescent fascination with arachnids and reptiles.
Yeah, what's that, Gene?
That's the mom-4U.
Air guitar riff.
Uh-huh.
Oh, I see. They have interesting names. DC-10 is the aircraft. Yeah.
Right.
Total lack of recall for dates is the oops gene.
Mm-hmm.
I have two comments about this paper. One I put in the show notes for you guys. It kind of frustrated me that they use this terminology throughout about genetic variation in chromosome Y influencing this, when I thought they should have said genes with variation among strains on the Y. It just made it sound like the Y chromosome was the whole, it was the whole Y chromosome, and really it's just gene variation. And the second thing is, I finally found a paper by Joe Netto, and I don't find where these mice have been published, their constructions specifically for the ones we used here, or they used here, are published. But this paper describes it pretty much as we said, at least 10 sequential back crosses with progeny genotyping at each generation, followed by intercrosses to homozygose the substituted chromosome. So an inbred strain is selected to serve as the donor for the chromosome substitution, and another is the host. They're intercrossed. The resulting F1 hybrids are back-crossed to the host strain. And then the progeny that inherit a non-recombinant chromosome are identified. So they go for something that has the whole chromosome and use that for the next back cross. To identify mice that carry a non-recombinant chromosome, they use DNA markers near both telomeres to make certain that the ends of the chromosome are transferred. Several intervening markers are also used to select against mice that might carry double crossovers and other complex chromosomal exchanges. And then to homozygose the autosomes and the X chromosome, they make the intercrosses between these heterosomic mice at the 10th generation backcross. And then they also sometimes will substitute in the mitochondria so that they have the mitochondria from the particular strain of interest. And then once they have these mice, they can maintain them by brother-sister matings.
And as we already discussed, I think in the case of making a chromosome Y consomic strain, I think it's more straightforward because there's no opportunity for the Y chromosome to cross over with another Y chromosome. You're going to get the whole chromosome every time. I would imagine you'd want to check to make sure when you're done, just in case something silly happened. It's interesting to me to consider, what the global effects are, and I guess we're seeing some of this here, of taking a Y chromosome out of some strain out there and sticking it in a new strain. I mean, I don't know how much variation there is in the Y chromosome, but it's an interesting concept. It makes me kind of uncomfortable.
Kathy, is there any sex difference in susceptibility of mice to adenovirus?
For mouse adenovirus, there's nothing that we've ever seen, but we haven't really focused on that. And that's something that's come up, most recently in NIH study section review guidelines, where there's more of an emphasis on, are you looking for sex differences?
Yeah.
And it's part of the reason why I asked Sabra Klein to come give a state-of-the-art lecture at ASV this year. So she is doing that. So
cool.
This, in a very broad sense, also points out, once again, for the lay folks out there, how important it is to, if you're going to do a clinical study or something like that, to include both males and females.
Yeah.
And in fact, even, a laboratory study, I don't know, you guys are more familiar with doing studies in mice than I am, but it seems to me that a lot of studies you're going to want to start out and do both sexes to see if whatever phenomenon you're trying to observe is the same in both sexes.
Right.
Speaking of, go ahead.
The limiting factor there is money and having enough to get enough N of both sexes to have statistical significance. And
so do people typically use just one sex so that they will get a uniform and just state that so that they'll get a uniform result? The sex issue won't complicate the interpretation?
That's often been the case. And so for a lot of the things that we've done with mouse adenovirus, we've done it with males because that's what historically had been done and published. Mm-hmm. And that's also historically because males are cheaper in general because females can be breeders, so they're worth more. Mm. But then when you start to breed your own, you get males and females, and you don't really want the females to go to waste. So it's a challenge.
So when we made a mouse model for polio, no one had any experience with that. So we just did half and half, right? And we didn't see any difference. Since we're talking about mouse models, so my lab spent many years working on polio in mice. And recently we have a interesting mouse model for Zika. And we had a grant go in, which the main criticism, what's the relevance of a mouse model to Zika virus? That was the main criticism.
I can tell you horror stories.
Yeah, I know. I've seen them in other cases, but
well, the response is you'd love to do the study in pregnant women, but there were some issues that came up.
Well, it's just that. Yeah, we can go back.
Come on.
But
what are you supposed to do this study?
This takes time, and the cycles are long. And I know that many people have had trouble with animal models, but I can't believe this is still going on, especially where there have been 500 papers on Zika that use mouse models.
Yes,
right. It doesn't make any sense. So you have to assume that the people saying this don't know the literature or something else. Let's do some email. We have many here. This first one is from Jessica. Good afternoon, beacons of viral education. Episode 424 was a contemporary favorite. I'm writing to win, but also in regards to the discussion of prion diseases. In episode 424, it was said that variant Creutzfeldt-Jakob disease prevalence was rare in the population, but I wanted to ask, is it? The fact is, given the potential long duration of the pathogenesis of the disease and the fact that we do not routinely test the expired for presence of variant CJ unless they were symptomatic, I think it's a pretty big assumption that it's rare. I think the newly developed assays are going to be extremely valuable in contributing to the prevalence numbers in given populations. Perhaps the rate is low, but we will hopefully find out once the non-invasive assay is refined and approved. Even then, the sample size may remain small because without a cure or therapy, people may not elect to be tested. I think that's a good point. We'll see. If you look at the epidemiological curve of symptomatic cases, yes, you're right about that. It's gone way down. And there was a clear epi curve for the outbreak, first in cows and then the variant disease in people. And we'll remind everyone that variant Creutzfeldt-Jakob was acquired by ingesting BSE-contaminated beef. Now and then there's still random cases here and there from people with apparently very long incubation periods. It was just one a couple of years ago, which I wrote about on my blog. So I agree. I think it will be interesting to know if there are some other cases out there, asymptomatic or otherwise. Okay. And in regards to the containment question, thank you, Alan, for asking. As a biosafety professional, it makes my heart sing to hear such important researcher safety questions raised. In my job, we discuss this very issue of appropriate handling practices involving human brain tissues with microtomes or cryostats. Considering the paper, I thought you'd covered this last year before. Let's see the link to this paper here. Not found. Well, that's a good paper. We did do that. Page not found.
Page not found. Oh, I've definitely seen that one before.
Which paper? Anyway, she thinks we did it last year.
She summarizes it.
Demonstrated transmission of prion disease through the aerosol route. It seemed unnerving not to play it safe and recommend containment for this activity, especially knowing that the true prevalence of variant CJD in the population is unknown. Aerosol transmission through the aerosol route. Does anyone remember that? That was apparently a PLOS Pathogens paper.
I am just fixing the link.
I got the paper. Aerosols transmit prions to immunocompetent and immunodeficient mice.
Does anyone remember if we did this?
Unfortunately, I don't remember.
Aerosols transmit protein.
I don't remember doing this one.
It doesn't ring a bell.
No, we didn't do this paper, but when we were talking about prions, we were talking about the biosafety level and
Right.
would aerosols be an issue?
All right, so they made aerosols with an aerosolizer, and they could transmit these infections to mice. Okay, yeah. In any event, I love the discussion, and as a previous field research technician responsible for removing deer brain stems with an apple corer.
That's a great image.
To test for CWD, chronic wasting disease, prions have been an interest of mine since the days of my undergraduate career. Okay, now we're taking an apple core and we are thrusting it into the brainstem of a dead deer. Is that correct?
That's
I would
what
assume
we're doing. Sounds
if
like
it's not a dead deer, it's going to be real soon.
Wow. I hope I'm number 17 because I love the topic of infectious diseases as much as I love traveling to far-flung places and eating weird things. The way I look at it is the cultural, social, and potentially medical adventure I may go on will add to my varied and interesting life story either way, and worst case, maybe make for an interesting epitaph. Wow, that's one way to look at it.
Yes.
Or be a case study on TWiV.
Yes.
In any event, I love the show and wish there were more time in a day, week, etc., so I could consume all episodes of the whole suite. I especially love listening to such provocative content when even NPR nauseates me for reporting on, and I realize they have to, the flagrant stupidity of the newly appointed administration. You don't have to repeat that aloud.
But
Thanks,
we did.
Thanks again,
Too late.
Jess. P.S. In Rhode Island, it's 42F and 59% humidity with a gale warning in effect. P.P.S. I wish Blue Apron had a holiday giving program where you could order meals for families in need. Food deserts.
And in fact, they do have a program at Thanksgiving that sounds a lot like what Jess is suggesting.
Cool. Speaking of travel, do you hear, comrades, about this considered ban on laptops from Europe to the U.S. on
I
airplanes?
can't believe that. That just is they can't possibly.
So let me ask you, given that, let's say this went through, would any of you purposefully not travel because you'd have to go reading a paper book again?
No.
No, you would travel? You have no problem with that?
Would the ban encompass iPads?
I think it's just
I don't think so.
laptops.
They're talking about larger devices like laptops. Plus, as a retired guy, I quite often travel without opening my computer, and a paper book is fine with me. Or
But
and an iPad.
there are an awful lot of people traveling on business who need their laptop because it runs the software for their sales application or their whatever it is, and they're going to be on a 5+ hour flight across the Atlantic, and that's going to be a major problem. I mean, they already implemented a ban like this for Arab countries, I think.
And it's all electronic devices there. I think it's iPads and phones and everything there.
Oh, okay.
See, I get a lot of work done on a transatlantic flight on my computer, and I would really not want to sacrifice
It's an idiotic measure.
Mm-hmm.
It's
going to get so much. It's going to get so much pushback from the public and from the airlines. I can't imagine this going through.
Well,
not to mention maybe the lawmakers.
I would have said that about a bunch of other things recently too. So,
Alan, can you take the next one, please?
Sure. Let's see. Ted. To the wonderful faculty of TWiV. Recently, mention of Dr. Richard Kessin's book, The Famine of Men, whose main premise is that a virus has made all males sterile while not affecting women, brought to mind the late crime novelist P.D. James' uncharacteristic foray into science fiction, the dystopian novel The Children of Men, which was also a very good movie, by the way. The James story is set, and Kathy has helpfully written that she wrote this in 1992, the book. The movie was, 08 or something?
Didn't see it.
So it was an early 2000s good sci-fi flick. The James story is set in England in 2021, and its premise is that due to universal infertility of unidentifiable etiology, no babies have been born since 1995. One female, who's a member of a dissident political group, finds herself pregnant, and the plot revolves about the small band's flight for survival. The early pages of the novel contain the author's well-crafted take on Western science's achievements, which I think you'll enjoy. Quoting, "We're outraged and demoralized less by the impending end of our species, less even by our inability to prevent it, than by our failure to discover its cause. Western science and Western medicine haven't prepared us for the magnitude and humiliation of this ultimate failure. There have been many diseases which have been difficult to diagnose or cure, and one which almost depopulated 2 continents before it spent itself. But we have always, in the end, been able to explain why. We've given names to the viruses and germs, which even today take possession of us, much to our chagrin, since it seems a personal affront that they should still assail us, like old enemies who keep up the skirmish and bring down the occasional victim when their victory is assured. Western science has been our God. In the variety of its power, it has preserved, comforted, healed, warmed, fed, and entertained us, and we have felt free to criticize and occasionally reject it, as men have always rejected their gods, but in the knowledge that despite our apostasy, our creature and our slave would still provide for us the anesthetic for the pain, the spare heart, the new lung, the antibiotic, the moving wheels, and the moving pictures. The light will always come on when we press the switch, and if it doesn't, we can find out why. That's good.
Yep.
Yeah.
Thank you all for the wonderful podcasts. May you keep the lights on in good health for many years to come. And Ted is a dentist at Nova Southeast University in Fort Lauderdale.
I did see this movie because my son wanted me to watch it because he said it has really long takes, right? Like have 10, 12-minute straight takes where the camera doesn't stop, which is really unusual in filmmaking, right? Now,
and very hard to do. Yeah.
Yeah. And they had to go through incredible lengths to hide the cameras. There's one scene when a car full of people is driving down the road and people are attacking it and it's being shot at and all this. And you get multiple shots by the same camera, which they put on this weird gimbal of some sort, and you never see it in the take. It's really amazing. So, you know, the director had this goal of doing long takes, which is for an actor is also hard to do, right?
Right.
Yeah.
And they had to do all this technical stuff. But the story itself is great, though. Yeah. There's this one miraculously pregnant woman. Yeah, it's great.
Yes.
It's really good. But too dystopian. It's very sad.
Oh, very dystopian, yes.
Kathy.
We're on to Kate.
Kate
writes, "Hi, TWiV folks. First time writing in after listening for about a year and a half and hoping I'm the lucky winner for the book contest. I fell behind a few weeks, so I was pleasantly surprised while listening to this past Sunday's episode to hear you still hadn't reached number 27. I hope I'm it. While catching up, I was excited to find the previous episode, number 429, was taped with a group from the University of Wisconsin. I am from Madison originally and graduated from UW in May of 2014. During my last 2 years, I was lucky enough to be an undergrad in Andy Maley's lab, influenza transmission, and it's great to hear about all the cool work going on in virology back home. I also just wanted to say thanks for all you do. Your podcasts help me get through my long days as a lab technician while I patiently wait for my boyfriend to finish up his PhD in microbiology. Soon it will be my turn to go to school. I'm applying to virology PhD programs this fall, and TWiV keeps me up to date with the ever-evolving field of virology. Thank you for the intelligent discussions and laughter. Cheers, Kate. In case you are still pulling, I do listen all the way to the end of the episode."
And sometimes beyond the end. There's good stuff. You should check it out.
I think that's what she means, beyond the end.
Rich.
David writes, "Hi, TWiVniks. It seems like every week I have an idea to write you about but never get around to it. Do you have a round to it to spare?" We'll come back to that. Anyway, this topic really strikes home, so I am extra motivated to contribute. With respect to your discussion of potential protein folding disorders that have non-neurological consequences, TWiV 431, there is at least one class that I am personally aware of. Desminopathies are caused by mutations in the gene that encodes the scaffold protein desmin, a component of the muscle contractile apparatus. At least in its autosomal dominant forms, desminopathy results in the accumulation of misfolded aggregates in muscle cells that eventually produce what is called myofibrillar myopathy. People with desmin mutations can present with muscular dystrophies that have varying ages of onset and rates of progression. The one in my family has a 4th to 6th decade of life onset, usually appearing as limb girdle type dystrophy, affecting the use of legs and more slowly the arms. Loss of ambulatory function sometimes may not occur until the 8th decade. Perhaps the slow progression arises because garbage disposals in cells can deal with the misfolded aggregates until they become too abundant or the cells get too old. Desmin is particularly abundant in cardiac muscle, and desminopathy can manifest as both cardiomyopathy and conduction disease. Sometimes the consequences of the latter, before any skeletal muscle issues surface, are the first sign. Sudden cardiac death can result. Other organ systems in the body that depend on muscle function can also be affected. I wonder if the cellular turnover or lack of same in a tissue is critical for whether it's likely to be affected by a protein folding disorder. Cells that turn over rapidly might not allow accumulation of the aggregates. On the other hand, cells like neurons and maybe muscle, when there isn't a lot of turnover, might be more susceptible. If this hypothesis is correct, one might expect exercise to slow the progression. I don't think the relationship between desminopathy and progression and exercise has been examined. This is counterintuitive, don't you think? Because you might predict that constantly breaking down muscle and trying to rebuild it in a person with a muscle disease would be deleterious, not favorable. So much for this diversion. It is currently 63 degrees, 17C, with cloudless skies in Hershey, PA. We're expecting 1 to 3 inches of snow after midnight. Don't you just love March in the temperate zones? Virology rules, and you folks are the court royale. Best regards, Dave. And then we've heard from David before. David Spector is an emeritus professor at Penn State Hershey.
So you have to be careful about this exercise. I don't know if that's been tested by clinical trial, but with chronic fatigue, they did a trial called the PACE trial, which the authors concluded that exercise was good, but it turned out that the results were bogus.
Yeah.
Interesting thoughts, though, and I think this goes back to, I think we had a conversation in TWiV 431 about where these where the genes encoding misfolded proteins are expressed and why it should be specific for one tissue or another. And David has some interesting thoughts about that here.
Yes.
Yeah, that's cool. All right.
And Vincent somebody has helpfully included a picture of a round to it.
Yes, a round to it.
There's actually a Wiktionary entry
about round to it.
Yeah, right. Play on words, reinterpreting the idiom to get around to it as to get a round to it. And there's
TUIT, yes.
Right. So it's actually a circular object. Oh, this is good. A circular object giving its owner the ability to get done anything that would have otherwise been put off to a later date.
Okay.
All right, I have an email from John Udell, who is a scientist at NIH and who has been on TWiV. This was sent on January 3rd, which I lost because he sent it to my Columbia email, and I don't have a good system of looking through that for TWiV emails. I have TWiV goes to Gmail, and I can have things come in and go into the TWiV folder automatically, and that's great. So, sorry, John. Many thanks to you and the TWiV crew for another fantastic year of podcasts. Now for the pedantic comment. In TWiV 422, there seemed to be a consensus at 43 minutes that the CNS is a dead end for viruses, in this case Zika. Rabies provides a clear example where CNS infection plays a critical, albeit indirect, role in transmission, since the increased physical aggression driven by the virus, which can even be seen in humans, though not in this gruesome video, fosters transmission through biting. As viral replication peaks in the salivary glands, it's plausible that CNS infection can foster transmission of mosquito-borne viruses through numerous mechanisms. These include changing host behavior, making the patient less likely to prevent mosquito bites, thus favoring transmission, modulating the immune response. The CNS plays an underappreciated role in systemic antiviral immunity. See attached papers for participation of sympathetic nervous system and flu immune response, and like herpes viruses infecting nerves that deliver the virus to barrier tissues, e.g. skin, mouth. The TWiV panel can probably come up with other potential mechanisms in a blue sky discussion. Yeah, I don't think people biting people is a very common mechanism of rabies transmission. Now, it may happen in animals, but we were talking about humans. And these other issues that John brings up, yeah, it's possible, but they're just plausible at this point. So I still stick to the CNS as a dead end for viruses in humans, unless shown otherwise by future studies.
Did you look at this video?
I did not. I don't want gruesome videos. Sorry.
Yeah, it's unpleasant. It's a documentation of an individual dying of rabies over several days.
No, thank you.
All right, a proposal. How about a father-son TWiV episode? Would be a first. My son Teddy is a postdoc at Sloan Kettering, though a virologist only by inheritance. Is that on the Y chromosome? I guess so. He's working on B-cell responses and using flu as a model antigen. We just published our first co-authored paper. This could be really fun and provide a new perspective on scientific careers. Very best wishes for the new year, John. Well, we did have a father-son TWiV.
We did.
Yeah.
Yeah.
TWiV number 7, I think. Let's look.
Was that when you had your son?
Oh, you
way back
had
there.
Yeah. My son, when he was very young. And he would I asked him once, he would never do it again. Now he's old, right? He wouldn't be caught dead with his father on a podcast. But anyway, I'd love to do it with you and your son, John. You could either do it in person here or we could do it by Skype. So let's see if John is listening, if he responds to that invitation. Now, let's do a couple more. Alan.
Karen writes, longtime listener, first-time caller. I've read about extracellular morphological changes in archaeal viruses, but can't find such a technique in any other domain. I thought I would ask the experts. Spreading the Twix word here at Stony Brooks.
Ken Stedman mentioned this last week.
Right.
And, there's archaeal viruses. They complete their changes outside the cell. I don't know of any others. Does anyone know?
Complete changes. Well, I mean, in terms of real morphological changes, no. But the final steps in maturation of HIV are extracellular, are they not?
Right.
Yeah, the retroviruses do some intracapsid changes. Yeah,
right.
But you can't see. It's not like you look at them and they're
not like a
different
shape
structure.
No.
Right, and if adenoviruses do some cleavages, but again, you wouldn't see those. Because they're internal proteins that are getting cleaved.
Right. Actually, now that you mention it, the phage T4 head changes size and shape during maturation because of internal cleavages. But I think that's intracellular. I think that's before it's actually released. Once it gets outside, it's done, I think.
Kathy, can you take Anthony's?
Yes. He writes, I've been thinking of this in the letters to TWiV 425. Where the question was, my question for you and your many guests is, what are the anomalous and unexplained phenomena in virology today, the facts and circumstances that defy explanation by the standard model? And he gives a link to the letters. And then says, before giving an answer, I'll have to be very confident of knowing the standard model. A few things did come to mind. If memory serves me correctly here, Sam Walton notes that in the early days of computer-generated sales reports, he'd go into the office at a quiet time, 2 a.m. Saturday morning. He placed the stacks of big fanfold printout paper on the conference room table and then walk up and down looking for outliers. When he found a store doing better with some item, Sam Walton would call the manager to ask what they were doing. Somewhere I read that the SEC trolls for insider trading by casting out an algorithm for every stock quote to swim through. The software nets behavior that does not mirror the market. If there's later something on some public announcement that explains the previous up or down movement, the SEC considers investigating. Clearly, insight and intuition will be self-seeding in the garden of the prepared mind. Even so, might there be devised automated nets for fishing in the growing waters of metadata. And before anomalies can be caught, the trend needs to be known. Information overload equals pattern recognition. At instant speed, the hidden becomes plain to see. And that's a quote of Marshall McLuhan. He gives a link to that. All of that is for what it's worth from Anthony.
The standard model. That's the point. You have to know the standard model, right?
Right.
Well, the first thing that comes to my mind is that in virology there is no standard model.
Yeah,
Right.
That's right.
They're all outliers.
Yeah.
You know, that's what's so fun about it, is that these things are wacky.
Yeah, you only have to teach an undergraduate class where you start to describe how things work, the principles of virology, and then you say, but there's an exception.
Yeah.
And that happens with almost every principle of virology that you talk about.
Rich, you're next.
Brandon writes, Dear TWiV Consortium, first, please forgive my formatting, for I'm not much of a letter writer. Second, my name is Brandon from Denver, Colorado, a longtime listener who has only emailed once. Even that was a half-hearted attempt to win one of your contests. As for the weather, right now, I look out from my desk to a somewhat overcast 18°C day, expecting snow over the next 2 days. We are long overdue for our March snowstorm, but I enjoyed the minus 22°C weather while it lasted.
I think that's not minus, but
I think it's a tilde. Approximately
about 22. Yeah, okay. I'm wondering where he's writing from. At any rate.
Denver.
Denver, okay. I write for no particular reason. A large part of it is the admiration I have for all of you. Part of it is that I have eked out just enough time to put together an email for you all, something I've wanted to do for a long time. My favorite episodes are TWiV 373 with Dr. Youngner and TWiV 395, The Cancer Thief. I have listened to those episodes many times over and have stopped me from remaining current. As much as I love the usual gang, your interviews with other distinguished virologists tend to be especially riveting.
Hey, listen, stop for a minute. That's funny because last week I said
the episodes that get the most listeners, right,
are not those.
Right,
goes down with the interview. Although some people do like them after all, so it's a mix, right?
Yep. Now a little about myself, at least the interesting part. The least interesting part, I assure you. I am a high school dropout from a family of high school dropouts that eventually realized school is cool. I went back and finished my high school diploma, and I'm now in my final semester as an undergrad, earning a degree in biology with a minor in chemistry. I am proud to say that I will be going to the University of Florida to pursue a master's degree in microbiology this upcoming fall, something that has never felt real or possible. I often have felt like quitting everything, but the good doctors of TWiV help keep me motivated. Through your constant humor, or humor from Dixon, stimulating discussions, and continued desire to learn. Thank you all for the work that you do. Tiredly yours, Brandon. How nice. P.S.
Well, that was the most interesting part, Brandon.
Yes.
That's awesome.
It is.
Yeah.
I do find Dixon quite funny and appreciate his attempt at the over 9,000 meme. I often find myself rolling my eyes with a smile on my face after one of his puns. P.P.S. There is a regional conference for ASM on April 15th, and I'm excited to attend, and I thought others might be interested. P.P.P.S. I miss Rich.
Aww.
That's
He's
nice.
back.
He's back.
You're back.
I've been gone. I'm back.
Yeah.
Now you're going to miss Dixon.
Right.
Yeah.
Yeah.
I think this is a quote that we can put on the front page. Where is it? I have often felt like quitting everything, but the good doctors of TWiV help keep me motivated. That's great, right?
Yeah.
This is why we do it, Brandon.
And if there are TWiV listeners who haven't heard it on TWiP, that's where the over 9,000 meme came into play.
Yeah, we had a case where the eosinophils were over 9,000. And I asked Daniel, what was the eosinophil count? He said, it was over 9,000. Dragon Ball Z, you know what we're talking about. Okay, let's...
by the way, I just want to point out that for the other undergrads and maybe graduate students out there, I have at times felt like quitting everything. You know? You do hit a wall now and then, okay? But it all worked out.
When I feel like quitting, I just yell at Dixon.
You know what I found? This is interesting. What I found is that this is in a sort of a microcosm. This is more not, but when I'm most frustrated is that when I don't have an idea for what to do next, when I don't have a hypothesis, okay? If I've got some kind of a plan, I'm okay. even if I know in my heart that the hypothesis is all wrong. But when I don't even know, have a clue what to do next, then I get discouraged. Mm-hmm. So just make something up. Get a hypothesis. Move forward.
Well, on that note, let's move on to some picks. Alan, what do you have for us?
I have a little press release and an associated video about a cool engineering project from this Chalmers University in Sweden. And
I just saw this picture.
Yes.
Right.
So Rich and probably Vincent will appreciate this. That the big thing in international yacht racing these days is hydrofoils. So they take these 90-foot catamarans and monohull boats. They put a deep keel on it with a hydrofoil, like an airfoil but underwater, and that allows those boats to fly. They lift up out of the water at a certain speed and they go screaming across the waves. So these engineering students decided to take a small one-design sailing dinghy called an Optimist, which is it's one of the most popular boats ever designed. It's used to teach kids sailing. It's if you didn't learn to sail on a Sunfish, you probably learned it in an Optimist. And it's about 8 feet long. So they took an Optimist and they put hydrofoil on it.
That's cool.
And they got it to work. And there was a huge amount of engineering work that had to go into this because, of course, this boat was not designed to do this at all. But it's really fun. And the video shows it successfully foiling into the, lifting up off the water and screaming across the harbor. It looks like a blast.
The video is really cool.
Yeah.
My kids learned to sail on an Optimist. I used to own 2 of them. The thing about Optimists is you buy them and from someone else used, and your kids learn and they outgrow them. Then you sell them.
And then you sell them,
else.
right?
Yeah. You go to any yacht club, you'll see all the for sale signs on the walls for Optimists. Yep.
Now, personally, I think a foiled Optimist should be called a pessimist.
Excellent. Which, by the way, is a great red wine if you should ever find it. It's called Pessimist. Pessimist. Remember that. It's really good if you can find it. Rich, what do you have?
I have chosen a NASA site, a site on NASA for a program that they have called HERA, Human Exploration Research Analog.
Is this where you're going next?
No, but I have a friend there. That's why I picked this. Okay. If you look at are you looking at this?
Yes, I'm looking at it. Mm-hmm.
So under the thing that says Human Exploration Research Analog, there's a picture of the 4 guys on the mission. Mm-hmm. The guy on the left with the curly hair, that's his name is Mark Settles. You remember my buddy Nassim? Mm-hmm. That's his son-in-law.
Oh.
Okay, and he always wanted to be an astronaut, but this is the closest he could get so far, anyway. So, NASA has a bunch of analog missions, and these are exercises that they cook up to simulate extreme environments of one sort or another, or extreme conditions that you might find in space exploration, and test them out. Either, test where you can do experiments here, test how the individuals do. And this particular one, HERA, Human Exploration Research Analog, they have a very small module, call it habitat, all right, that simulates a spacecraft, and they have over a period of years been experimenting with longer and longer isolation in this thing, and they're on campaign 4 now, and this is mission 1 in campaign 4, and they're up to 45 days. So these guys are going to go into this module for 45 days, and they have, a bunch of experiments and stuff to do. But the biggest experiment is how they get along with each other for 45 days in this
they're locked into this. And some of these, they apply some kind of a delay to communication
I don't know whether that detail applies.
Like you can't just sit there plugged into your iPad and getting your latest updates.
Right.
I think they do a lot to really make it like a realistic you're on Mars kind of experience.
And if you flip through this, they've got from some older missions, somebody giving a tour of the habitat and that kind of stuff. It looks pretty interesting. I was wondering as I was looking at this, how they screen the individuals to participate. And I was even wondering whether some devious mind might actually pick out personalities that look like they would qualify to do this, but might not get along with each other.
Maybe.
And see how that works out,
That's pretty cool.
Mm-hmm.
Well, I never wanted to be an astronaut, so I'm not going to do that.
Could put together some candidate lists for people who ought to be locked into this together.
Kathy, what do you have?
I picked something that's kind of along the lines of something we talked about last week. So it's a continuing arc about unrecognized privilege, and it's science because it's social science research, and Shankar Vedantam has not yet talked about it on NPR, to my knowledge. And the idea is it's from research here at the University of Michigan, so I saw it on the news blog page. The gist of it is that the transition to motherhood is difficult for all new mothers, but there are some hidden advantages that many middle-class moms chalk up to luck, and then people who are less than middle class aren't so lucky, and so how that plays out in terms of motherhood. But she found a lot of women describing themselves as lucky if they had parental leave, or they could get time off work, or they had family members that could provide free, high-quality childcare, or if they had an understanding boss, and so forth. And then when you think about it, there's a lot of new mothers who aren't so lucky, and that's disproportionately low-income women.
Mm.
Yeah,
Right,
this is where the word fortunate works better than luck.
Yeah.
Or privileged, or
Right.
because it really is. These are not random things. You were able to take time off work because you are upper middle class and work in a place that allows that, and because you're a salaried employee. If you were making $8 an hour at a parking garage, then you wouldn't be able to do that. So yeah.
Right. And there's a big discussion in this about breastfeeding and how it's a real challenge for all working mothers, but it's especially challenging for low-income mothers who, don't have a way to pump at work and, have a refrigerated way to keep the milk and all those sorts of things. And so it's just another way to become a little more conscious about privilege.
Yeah.
Unfortunately, the current climate just doesn't appreciate that at all.
No.
Right.
Unfortunate. It's not going to get better anytime soon. Well, I have two picks. One of them is entitled "Make Measles Great Again in Michigan." So I thought Kathy would sigh at this, right?
Yes. Oh, yes.
So this is a nice article in Science-Based Medicine, which is a blog I like. It's by David Gorski. And
David goes at some length, so buckle yourself in, but it's worth at least reading through this and seeing what all is going on. And
this is a long article, but it's very good. And it's all about, the attempts by the legislature in Michigan to roll back some of the barriers to vaccine exemptions. There's this sense on the part of lawmakers that we've gone too far. Make it too hard for parents to get exemptions to vaccinations, which of course is preposterous, right?
Yes. And
so this describes it all. So I think you must be familiar with this, right, Kathy? Or not so much?
And the pandemic symposium that I went to about a month ago now had two people in public health, one at the state level and one from Detroit. And I can't remember which one of them said it, but they quoted where Michigan is in the unvaccinated ranking. And Michigan is 43rd in childhood vaccine completeness. And it's not all due to anti-vaxxers, that person said. Mm-hmm. But 43rd is pretty bad.
What are the other reasons for it? Was it mentioned at all?
Not really.
Okay.
Not really.
What is Massachusetts, Alan?
I don't know.
I think you're pretty good, though, aren't you?
Massachusetts actually does pretty well with this. In order to get a vaccine exemption, it takes some effort. You need to go to your doctor and have your doctor say that your child shouldn't be vaccinated, which is a much higher hurdle. At least I think that's what it is. I remember seeing something and saying, good.
Well, and that's from reading this link that Vincent gave us, that's essentially what the situation is now in Michigan, is that you have to go and talk to a doctor. But the legislature has before it a proposal to get rid of that.
Yes.
Why? Why would you do that? What's the justification, right?
Because you have a bunch of anti-vaccine people telling you that this is taking away your freedom.
Right.
It's a very, very heavy, I can't think of the word, propaganda sort of thing.
Yeah.
Right.
All right. On a lighter note, I have an article in the Times. Which tech giant would you drop? The big 5 tech companies increasingly dominate our lives. Could you ditch them? So they have this lovely interface where you're given 5 buttons: Alphabet, which of course is Google, Apple, Microsoft, Facebook, and Amazon. And in what order would you ditch them? Okay. So my order, I want to hear yours. My order was first Microsoft, second Facebook, third Alphabet, fourth Amazon, and finally last Apple.
Wow.
What about you, Alan?
Wait a minute. Vincent, what did you say again?
Microsoft, Facebook, Alphabet, Amazon, Apple.
That's impressive. I had Facebook, Microsoft, because I've already ditched or not used either of those. I mean, that's status quo. Amazon, Apple, and Alphabet.
Google is the most important to you, huh?
Google would be the hardest to get rid of. I've got a Gmail email address. We use Google Docs on TWiV. I Google search things all the time. There are other search engines, but that's the default.
Yeah.
And just very hard to get rid of that.
How about you, Rich?
I'd get rid of Facebook first by an order of magnitude.
Yes.
I said Amazon second, but with great trepidation. I said Apple third, only because over the years I've been conditioned to Microsoft against my wishes. Microsoft fourth, and Alphabet for basically the reasons that Alan describes.
How about you, Kathy?
Well, Facebook would be first because I'm not on Facebook, so that's no problem.
Of course.
Probably it's a toss-up between Amazon and Microsoft. I mean, if I had to, I could finally really use Pages and,
Yeah.
and then Alphabet, and then Apple.
So you can look at this article of what other people did, and the first dropped 56% said Facebook.
Yes.
Yeah.
Which is funny. So many people are on it, and yet they want to not do it.
Yeah.
That means it's addictive. Microsoft was next. Apple was third, Amazon, and then Alphabet. But dropped last, Alphabet was the leader, 39%, and Apple, 33%.
Interesting. This guy said that his last was Amazon, right? The last he would drop.
Yes.
No. Yes.
That's what he said, the author, right?
My reaction was, I can go to a store or something like that, but Amazon is a whole lot more than a store.
Yes.
Microsoft, Facebook, Amazon. I put Amazon fourth, but it could be third. I do use Gmail and Google Docs a lot, but they're replacements for those. Whereas I use my Apple products just about every minute of every day.
Right.
My office is full of them. I use them to be productive. I write, I make slides, I do podcasts, movies, and everything, and I just don't want to use Windows. So that would be pretty important for me.
Yeah.
Well, there is Linux.
I almost wish they would drop Microsoft, so that I could go back and use all Apple products again.
Yeah, that's right. You used to.
Because I really do like it. Yeah, I was an addict.
Anyway, it's fun. Check it out. It's a cool...
I like that.
Cool little survey there. I like that. We have a listener pick from Steve who writes, I just read this brilliant editorial view from Richard Horton. I think it would make a great listener pick and deserves wide circulation for pinpointing one of the most insidious and damaging trends of our time. Who knows, it might even help him see we don't just like harassing him over pace. It's called Difficult Truths About a Post-Truth World, published in The Lancet. This is a great piece from Richard Horton, editor of The Lancet. It should remind us all that while Brussels may seem to have more of a green, a socially responsible face than our own rabid government, the EU exists for the explicit purpose of promoting business and is run by the business lobby for the business lobby. Especially note how the principle of truth of scientific objectivity is being encouraged to be thought of as just another biased view from vested interests. This lobbying angle literally is capable of destroying worlds. All the best, Steve, from Luton Beds, England. Now, I had some correspondence with Richard Horton over the PACE trial. Right. This is funny because we asked him to retract the PACE trial for chronic fatigue, myalgic encephalomyelitis. And, of course, he said he would not, but at least he did respond. Although he said we would publish our letter criticizing it, but then he decided not to. Anyway, this is a very interesting article. You should check it out. And Steve is right. It's really good. From his perspective in England, of course, it's really we don't maybe think about this very much, but you should check it out. It's not very long. All right, that's it for TWiV 441. You can find it at microbe.tv/TWiV. You can find it at Apple Podcasts. If you have a favorite podcast player on your mobile device, you can search for it. You'll find it, TWiV, This Week in Virology. You can subscribe to it. Just subscribe. You know, you don't have to listen to every episode. Just subscribe so that you'll get it automatically. And if you're so motivated, you might listen. You know, if you're in space orbiting the Earth, you might want to listen.
Absolutely.
It can happen. Send us your questions and comments to TWiV at microbe.tv. Consider supporting us so that we can travel, but not back from Europe without our laptops, of course. We can travel and do TWiVs all over the place. Go to microbe.tv/contribute for how you can do that. Kathy Spindler's at the University of Michigan in Ann Arbor, Michigan. Thank you, Kathy.
Thanks. This is a lot of fun.
So mouse paper, it was up your alley.
Definitely.
Of course, virology is up your alley too, but this one especially.
Yep.
Thank you for your insight. Rich Condit's at the University, well, I should say, let me start again. Rich Condit is an emeritus professor at the University of Florida in Gainesville, currently in Austin, Texas. Thank you, Rich.
Sure enough, always a good time.
Alan Dove is at alandove.com. He's also on Twitter. Thank you, Alan.
Thank you. It's always a pleasure.
I'm Vincent Racaniello. You can find me at virology.ws. The introductory music on TWiV is composed and performed by Ronald Jenkins. You can find his work at ronaldjenkins.com. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral. Hey there, how are you?
Hi.
How are you doing?
Good.
Where are you, in Texas?
Yes.
Do you like Texas?
Yes.
Is it hot there today?
Yes.
Wow.
How old are you?
I'm 8.
Pretty good.
hey.
You speak beautifully.
Yes, you sound great.
Maybe you should be a podcaster. You don't know what a podcast is?
No.
That's what your daddy does, your granddaddy does. He records with that microphone there and does This Week in Virology. Have you heard of that?
Yes.
Oh, great.
Good.
Are you wearing headphones?
Yes.
When you speak, do you hear yourself in the headphones?
Yes.
Do you ever say no? Yeah, that's
How much
funny.
longer does school go on?
Until June 1st.
Oh, not too much longer. And then what are you going to do?
I don't know.
Oh,
That's the best way to be. I don't know.
yeah.
Yes.
It's okay because we don't know either.
Okay, Harper, say goodbye.
Bye, Harper.
Bye.
Nice name.
Bye.
Wow.
Bye, Harper.